Dr. Fillit is a medical doctor, a geriatrician and a neuroscientist. He's the founding executive director of the Institute for the Study of Aging and Estee Lauder Family Foundation and the Alzheimer's Drug Discovery Foundation, ADDF. These organisations have a common mission of accelerating drug discovery for Alzheimer's disease through venture philanthropy. Dr. Fillit has had a most distinguished academic medical career at the Rockefeller University and Mount Sinai School of Medicine, where he's currently a clinical professor of geriatrics, medicine and neuroscience. He is the author and co-author of more than 300 scientific and clinical publications and senior editor of the leading international textbook of geriatric medicine and gerontology. Dr. Fillit has received several awards and honours, including the Rita Hayworth Award for Lifetime Achievement from the Alzheimer's Association. Throughout his career, he has maintained a limited private practice in consultative geriatric medicine with a focus on Alzheimer's disease and related dementias. Founded in 1998 by Estee Lauder's family, the Alzheimer's Drug Discovery Foundation is a leader in funding innovative Alzheimer's drug research worldwide. ADDF follows a venture philanthropy model, funding breakthrough research in academia and the biotech industry. The ADDF is the only charity solely dedicated to funding the development of drugs for Alzheimer's in the world today. Through the tremendous support of donors, the ADF has awarded more than $95 million to fund over 500 drug discovery programs and trials in 18 countries around the world. Could I please introduce Dr. Howard Fillit? Thank you very much.
Thank you. Thank you very much. Yeah, thank you. Well, thank you, Samantha. Could you turn the lights up, please?
Oh, yeah.
Okay.
Just try to get more.
Yeah, you can make them a little higher than that, actually.
A little bit louder?
Yeah. Yeah, I like them bright. Thank you.
You just want them on?
Yes.
All right.
Okay, thank you. I like the lights so I can see everybody. I want to thank Bales and Samantha and Sarah for inviting us here today, and Helen for all of her efforts, and Bob in bringing us here today and giving us an opportunity to meet all of you and learn more about Australia in general, and particularly to meet some of the investigators that our foundation has been funding here in Australia over the course of the last more than 10 years, actually. It's a great privilege to be able to talk to you today about what we're doing. I am a geriatrician, and I really resonated with what was just said by the young investigators in terms of multimorbidity. You saw a funnel, and I'm going to show you a funnel. Everything I'm going to talk to you about today is related to how we translate gerontology, the biology of aging, into new drugs for Alzheimer's. I'm going to include amyloid, but I think that we're going to be discovering new drugs for Alzheimer's for the next 100 years, just as we've been discovering diabetes drugs for 100 years and hypertension drugs for about 80 years. Aging is the leading risk factor for Alzheimer's disease. You heard a bit about some genetics, ApoE4, and in fact, although I don't have a slide of this, we are funding two programs, one at Harvard and one at Cornell in the States, to use a virus to introduce the ApoE2 gene, which is protective, into people that have ApoE4 to try and reverse that defect. And we have other portfolios of trying to correct the genes. But generally speaking, aging is the leading risk factor. And in fact, aging is the leading risk factor for many diseases of the geriatric population. And you can see here in this slide, actually, that all diseases actually increase with aging. We all are going to suffer diabetes, hypertension, cancer. We think of breast cancer as a middle-aged disease. In fact, breast cancer increases with age, and so on. So actually, Alzheimer's is kind of interesting because you can see here that while these sort of have a straight line with age and begin in middle age, the expression of Alzheimer's clinically really is not beginning until there's some delayed onset here in the 50s and 60s when you heard about some very early onset, very rare, really unusual cases, mostly people that have a genetic form of Alzheimer's, but not the sporadic form that we associate with aging. So in my mind, trying to translate the clinical information into drugs through the biology of aging, what this tells us is that the neurons are actually quite interesting in terms of their ability to protect themselves from the damages and the ravages of living on Earth. And I do think that living on Earth has a great impact. If you take a car and put it in a garage and you don't drive it, in other words, you don't use it, it's still going to rust. It's still going to live under the sun and in an oxidative atmosphere, and it's still going to degenerate, even though it's just a car. And I think our bodies do get affected tremendously by living on Earth over the course of 80 years. The thing is about the brain is that as we lay down neurons, we don't want our neurons to proliferate. We want them to settle down, create circuits, and then remain there for years. So when we learn to tie our shoes when we're 3 years old, we want those cells to hang around for 80 years, whereas our skin cells are dividing every day. Hundreds of millions of cells are turning over in our gut every day. And that's the reason why those cells, when they become abnormal through the ravages of aging and living on Earth, and genetics and epigenetics, when they become dysregulated, they become hyperproliferated, and they become cancers and tumors. But in the brain, when a brain cell divides, it sets it into a cycle called apoptosis, which is programmed cell death. The brain cells say, this is a computer, the circuits are laid down, and if I'm going to be damaged, I got to get out of here, and I'm going to just commit suicide and die. So when we see brain cells go into a proliferative state, we see them die. And so we want to try to understand the mechanisms of protection here, why brain cells are somewhat protected, how they protect themselves, and learn from the biology of gerontology to develop drugs for Alzheimer's disease. And for other forms of dementia, as you heard, vascular dementia, small vessel disease, Lewy body disease. In fact, I totally agree with what the proposition was. When we see people with dementia, we can sort them out with the brain scan that our foundation helped to develop, that you've heard about, this amyloid scan, to identify people that have beta amyloid plaques. But when you actually look at the pathology of old people who are demented, they have multiple pathologies. Almost more than half of them have vascular pathology. 35% have a Lewy body pathology. Some of them have what's called TDP-43 pathology. So this really is a case of multimorbidity and complexity at the pathological level and at the clinical level. And so when we translate that biology into mechanisms of understanding what they are so we can develop drugs, let's think for a second about what that funnel looks like. And the first is epigenetics. So living on Earth, the link, as I'll tell you about in a minute, between the environment and our genes are epigenetic changes. We lay down the DNA. It's the template for who we become and what our cells are. But then they're modified. Genes are not expressed. All the genes, our brain cells don't express kidney genes. Our heart cells don't express brain genes. And that's because of epigenetics and the way that the genes are regulated. And as the environment changes, the environment can affect our genes. Think about it, and I'll tell you about that in a minute. Let me just move on here. Another thing is we heard about the mitochondria. The mitochondria are the powerhouse of all of our cells. Every one of our cells has these powerhouses that are using glucose and oxygen every minute of the day to generate energy that we use. Now, the brain, as I'll tell you about, is very heavily dependent on energy. And with aging, this theory has been around. John Blass first proposed this in the late 1970s, that there was a mitochondrial energy failure in the brain that accounted for neurodegeneration and led to excess oxidation because the mitochondria use oxygen and create free radicals, and failed energetics. Another thing is neuroendocrine aging. Here in Australia, there's a company called Actinogen that is developing drugs to block the stress response. And we know, for example, that pilots that undergo a tremendous amount of jet lag and stress have smaller hippocampi. Their brains shrink. We know from biology that cortisol in excess is neurotoxic. We know from the biology of gerontology that in young age, the stress response goes up and comes down. But as we age, the stress response becomes dysregulated, and cortisol goes up, and then it stays up, and it's neurotoxic to the hippocampus. Also, estrogen, the time of the menopause, dramatic falls in estrogen levels this is still an ongoing field. It's one that's not very clear. There's not good evidence that estrogen replacement can protect against the brain ravages of aging in women, for example. But it's still an ongoing field, and there are other metabolic disturbances, such as thyroid disease, that are risk factors for dementia that we don't completely understand. All of our proteins, form is function. Form is function. We have the primary structure, and then the three-dimensional structure, and then the quaternary structure. And it's only when you get that three-dimensional structure that proteins start to act with each other in a way that creates biology. When proteins become oxidized or have other what are called post-translational changes, they dysfunction, they misfold, they aggregate. And this is going on with many proteins in our body. Amyloid, beta amyloid in the brain, is one of the key sine qua nons of this dysfunction, of this misfolding. But it's not just the misfolding that occurs that leads to these toxic misfolded proteins that can kill brain cells. There's a failure of, through evolution, our body has developed what are called chaperones. And all animals have these molecules that protect the proteins from misfolding because it's constantly happening, even in bacteria. And so evolution has designed mechanisms to create chaperones that prevent or treat misfolding. And that's a potential drug target. There are companies that we're funding right now that are developing better chaperones because the chaperones decline with aging. But also through aging, these misfolded proteins become the garbage of the cell. They're toxic to the cell. And the cell has mechanisms, the garbage disposal system, it's called the autophagic system, to remove misfolded proteins like beta amyloid, like tau, like TDP-43, like hundreds of other proteins. And with aging, the garbage disposal system starts to fail. So drugs are in development to try to enhance the garbage disposal system, the autophagy system, the proteostasis system, to prevent this accumulation of misfolded proteins. Inflammation. We teach medical students in geriatric medicine that the first sign of pneumonia in a 40-year-old is cough and fever. But older people, although they have a decline in their inflammatory response, the first sign of pneumonia in an older person is confusion. It's delirium. It's loss of cognitive function. And that's a good example of how systemic inflammation, which is a hallmark of aging, all of us as we age are experiencing inflammation. Our proteins, our joints are breaking down. There's inflammation, and it's affecting our brain. This is well documented in animal models and in humans. And so preventing systemic inflammation effects on the brain can be a therapeutic target for Alzheimer's. And then we have neuroinflammation. The very specific microglial cells that are specific to the brain play an important role not only in removing the misfolded proteins and the plaques and the beta amyloid, but also in creating toxicity because they release molecules called cytokines, which are cytotoxic. And so we have this double-edged sword that I'll talk about to prevent the neuroinflammation. We heard a lot about white matter disease previously. We're very interested in that. It's become very clear that Alzheimer's is also a disease associated with white matter changes, that people that have these deep white matter changes are also affected in accelerating Alzheimer's disease. The reason is the carotid arteries are these big arteries that come in over the brain, and then they dive deep into the brain. And as they dive deep into the white matter, they become smaller and smaller arterioles and then capillaries. And they're subject to ischemia. They're subject to hypoxia. Now, in the deep white matter is where we have these long tracts that connect the different parts of the brain, the different modules. These are where the connections live. This is the white matter. And the glial cells that are affected in multiple sclerosis, for example, these glial cells that make the white matter become ischemic. So it's not the neuronal bodies in the deep white matter that become ischemic, but the glial cells. And we see white matter disease in Alzheimer's pretty commonly, and blood-brain barrier dysfunction. You can't have a bad. And so all of this, we think, now the importance of this is that there's a reasonable chance that the anti-amyloid therapies will work. But what I've just described to you is the fact that there are many biological pathways to get to neuronal death. And I don't think any of us really feel that, unlike the genetic causes where you have a genetic defect in the amyloid gene that causes Alzheimer's, that that's going to be what happens in old age to the brain, and that the anti-amyloid therapy will be the cure. I don't think it will be. I don't think we think that. We are going to be discovering drugs along these novel pathways for the next 100 years. And that's an optimistic point of view, because we understand in the last 30 years more about the biology of Alzheimer's disease. And I feel confident to speak before you today and say that I think we know as much about the biology and the pathology and the neurophysiology of Alzheimer's disease as we know about heart disease and cancer. But we're 20, 30 years lag time because we didn't really start Alzheimer's research until the late 1970s, whereas we started heart disease research in the 1930s, diabetes research in the 1920s, cancer research in the 1920s. So we have this lag, and it's a really exciting time because we've developed the knowledge about the biology, and now is the time to translate that knowledge into new drugs. So let's talk about how we're developing new drugs. And I'm going to, I've given some sort of lay interpretive talks, and I'm going to try to interpret this for the lay audience, but I've tried to make this more of a technical talk, so I hope I can explain what I want to say here to everyone in the audience. What I want to do is spend a few minutes telling you how we develop drugs, because we think of medical research in terms of what is basically more often the biology, the basic science, the creation of new knowledge. But drug discovery research and drug development research is a separate field of science that's very fascinating, very technical, and very interesting. So I want to just tell you a little bit about what that is. So you have basic science, understanding the causes of disease, and then you have drug discovery, which is translating this knowledge into new drugs, and then a regulatory and pharmaceutical science period where we have to figure out how to deliver those drugs in a pill or in a biological infusion and know that these drugs are safe and that the FDA or the EMA or the Australian authorities will allow us to test these drugs in human beings. So a tremendous amount of regulatory safety and toxicology. And then finally, the specialty of doing clinical trials. Drug discovery can be isolated into various periods of time. So we first have identification of a target from the basic science, and then what's called screening, various ways of screening drugs, what we call phenotypic screening or rational biochemistry, rational medicinal chemistry. Lead discovery, that's where you find a molecule that becomes your lead compound in a program. You identify leads from some thousands, sometimes millions of compounds that are being tested. And then these go through a round of medicinal chemistry, the wizards of medicinal chemistry that can see a molecule on a page and tell you whether or not that'll be a drug, and if it's going to be a drug, how is it going to be commercially viable to synthesize that drug, and then finally the preclinical testing. Now, this chemistry thing that is the very basis of small molecule, we want to have a pill one day, is really amazing. We've known about aspirin, very interesting small molecule. Comes from the bark of a tree, really, right? We sort of don't completely understand how it works. But the universe of all small molecules is huge, 10 to the 100th, right? Greater than Avogadro's number. So the possibilities of small molecules that could potentially become drugs is essentially infinite. It's huge. And the same thing with biologicals. We know that there are roughly 20,000 human genes and 100,000 proteins. So one would say, there's so many opportunities to develop a drug. But the fact is that of all the small molecules and biologicals out there, there are only about 10,000 drugs in the FDA armamentarium that have ever been approved. And most of these, the majority of these, are variants on formulation and delivery. In other words, you have a pill, and it could be taken one way or another, extended release, immediate release. It might be a skin cream or a patch. It could be taken orally. So when you really drill down on the number of drugs that are actually out there, there are less than 300 out of those 20,000 druggable genes. Druggable genes means that you could potentially make a drug out of it. And in the armamentarium, fewer than 500 in all of the time that we've been doing research for 100 years on different diseases, less than 500 distinct chemical entities that are based on fewer than 50 what are called chemical scaffolds. So the medicinal chemist will take a scaffold of a molecule, and they'll put different groups on it to make a drug. And there's less than 50 scaffolds. So that's kind of interesting, I think. The other challenge is that with biologicals, a lot of genes, a lot of the proteins that we think of that we learned from the biology are beyond the reach of classes of drugs that are currently available. Small molecules have a hard time not only passing the gut and surviving stomach acid and then getting in the blood, but getting inside cells to work, to hit their targets. So we see this fantastic biology, as I'll show you in a second. But when you're trying to create a drug, can the drug actually get inside the cell? And not only that, but in the brain, in Alzheimer's disease, we have the added challenge of getting through the blood-brain barrier, which, in other words, through evolution, the human brain especially has developed this barrier so that stuff can't get into the brain and muck up that electrochemical system that is essentially our computer that allows us to think. A lot of times we get applications from people from scientists around the world that say, I really think this is an important pathway that I've discovered here, and I want to develop a drug against this. Well, there's some fundamental things that a drug discoverer would look at that a biologist wouldn't think about. Because, for example, GSK3 beta is an enzyme that phosphorylates tau and is thought to misfold tau. And for a long time, it was attempted to make GSK3 beta a drug target. But the problem is that in the cell, and this is a simplified version of how the cell works, right? You can see that there's all this redundancy around GSK3 beta. So if you inhibited GSK3 beta and stopped the phosphorylation of it, the cell has many different ways to get around it. So very often intracellular pathways, even though they're biologically validated to play a role in the disease, and we learn about that biological validation from our basic research, it's not necessarily a good drug target. And in fact, more often than not, targets that are at the cell surface that have many downstream effects are historically better targets. And you can see that in this slide whereas a biologist will describe many different targets to you and say, I have the most novel pathway for how Alzheimer's disease happens. A chemist or a drug discovery will tell you that this is the hierarchy of the possibility of how drugs could be developed. GPCRs and enzymes GPCRs are molecules that sit at the cell surface and have many downstream effects. And most drugs are actually GPCR molecules that affect the GPCRs in the cell. Enzymes can be inhibited. We can learn about the pocket, and we can get an inhibitor in there, and so on. Whereas protein-protein interactions, which I won't go into, are very difficult. And we get a lot of grants where people say, we want to inhibit this protein-protein interaction. For example, we want to use a small molecule to prevent the aggregation of beta amyloid into plaques. That's very difficult because the A-beta has many different points of contact when it forms those beta helical sheets. And to use a small molecule that's a pinpoint to try to inhibit these big molecules is not really a practical way to develop a drug. Another problem that we have in the field in developing drugs in terms of challenges is the animal models. We've cured animals with Alzheimer's disease 500 times now. We create these transgenic animals that express beta amyloid, and they're good for target engagement. They've been good to show that a drug hits its target. But they haven't translated into new drugs for Alzheimer's disease. And very often the rigor of the way these trials are reported is poor and not reproducible. And so the NIH, we've been working with the NIH to improve the rigor. And just as there's right now, for those of you who know about it, the clinicaltrials.gov site that's supported by the US government, so that all clinical trials in the world get registered publicly on a website that anyone can go to and see what trial is being done by any drug company. It's required for FDA approval. What's being developed now is a preclinical trials.gov site so that negative animal trials and the quality of animal trials can be reported. So basically, we need teams to develop drugs. And these teams can exist in big pharmaceutical companies, but they're not readily available in academia. And they're certainly not readily available in virtual biotech companies that are early stage, that have novel intellectual property that might have been spun out from a university. And so the way to develop these teams is a key issue in how we how we translate basic science knowledge into new drugs. And our foundation has been doing this for about 15 years now in terms of helping our investigators to access the teams around the world. We have a website called ADDF Access, which is kind of a match.com. I have a full-time PhD working on this in my office, where we have 6 PhDs, neuroscientists, that are constantly searching the world and helping investigators to develop their drug discovery science. And through ADDF Access, we've vetted about 150 contract research organizations that can help in creating these teams around the world on a virtual basis. So if you have a novel biological target and you want to do a high throughput screening and you want to buy a compound library of 2 million compounds and apply that to an assay, you can do that virtually. And that's a big model because it's very cost effective. You don't have to pay rent and light bulbs and overhead and administrators. And a lot of early stage companies that are high risk are virtual. And I would think that in a place like Australia, this would particularly be a very good model. Now, I've been doing this about 35 years, and when I started, there were basically no clinical trials going on. And we knew nothing about the biology. Today, there's about 100 different clinical trials. This is from a recent paper. We have about 24 novel drugs in phase 3, which is the last phase of discovery. Phase 3 trial typically costs anywhere from $100 to $400 million. You need about 2 of these to get a drug approval, maybe 4 sometimes. So we're talking about risking billions of dollars. Of course, this is mostly done in big pharma. A lot of these we've contributed to as a foundation. We're participating in this Navaldobine trial. I'll talk about that in a few minutes, and some of the others. We have 45 drugs in phase 2. By the way, of the 24 drugs in phase 3, 7 are symptomatic agents that are looking to improve on current drugs on the market, like Aricept and Namenda, and 17 are proposed disease-modifying therapies. And out of that 17, 75% are anti-amyloid. So the drugs in phase 3 right now are very narrow and on primarily the anti-amyloid target. It's not just the one that was just reported in Nature, but many of the companies are focused on anti-amyloid, beta secretase inhibitors, vaccines, and so on. That's good news in a way, because we're going to validate that target in a drug program in phase 3. But what about innovation? What about these other pathways? Where's the new drugs going to come from? And about 24 in phase 1. Some of these mostly anti-tau. We heard about this biomarker. I'm not going to talk too much about it, except to say that the first biomarker that came to market was one that was brought to market in part through our foundation's efforts. We were really the first funders of this program, where you can actually see in the red here the beta amyloid in a living human being through a PET scan. And we were the first funders of this program back in the year 2000. I'm not going to tell that story, but it really has changed the world in several ways. It's enabled early diagnosis. You heard from the clinicians about the unavailability of this test in Australia. It is available and approved in Europe and in the United States, and I do use it in my practice. And in the U.S., it costs between $3,000 and $4,000 U.S. dollars. But I do find it very valuable when people come in with memory problems, and I can tell them definitively whether or not they have Alzheimer's. And if they don't have it, that's very reassuring generally, because then maybe they have something like hippocampal sclerosis or vascular dementia, or maybe they have something that's treatable, like you heard about, like depression or polypharmacy, which I could do anyway, but I know that they don't have Alzheimer's pathology. If they have Alzheimer's pathology, then I can help with care planning, or maybe refer them into one of the clinical trials that's going on now, because they would qualify because they have a positive amyloid scan. So it has changed clinical care, but right now, Medicare doesn't, in the U.S., pay for this test, and there's a study of 19,000 people going on in the United States, sponsored by Medicare, to see what the impact of this scan is on clinical management of patients. And if this is called a CED, an approval basically with data development. And if it turns out that through this study that the scan is proven to have an effect on clinical management, then Medicare will pay for the scan. And we can also do spinal taps and get information. The other thing that we're going to hear more about is how the scan has enabled more rigorous clinical trials, because for the first time, this Biogen study that was just announced used the scan to enroll people who actually had Alzheimer's disease. and actually had beta amyloid in their brains. And that had a big effect, I think, on the outcomes and improved the rigor and the efficiency of these trials. And it's enabled prevention. And you heard a little bit before about how we can now scan people who are normal, who are 75, and that's going on here. Colin was telling me about it. In ABLE, this is the largest site in the world for this A4 trial, which is a trial studying people who are clinically normal but have amyloid in their brain to see if you treat them with an anti-amyloid antibody, if you can prevent the onset of symptoms from the Alzheimer's disease, because when somebody's got beta amyloid in their brain, they have Alzheimer's disease. They might not have dementia or mild cognitive impairment from Alzheimer's, but they have Alzheimer's disease. And this was the article that just came out, this aducanumab, which is a very interesting antibody. We could say from a drug discoverer's point of view that when you've seen one antibody, you've seen one antibody. And there's another antibody called solanezumab that's being developed by Lilly. And we might hear about that trial by the end of this year, and that will be also very interesting. Here's what the scan looks like. Here's the baseline. In other words, when people were entered, they all had amyloid. And here's the placebo group. After 1 year, you can see there was no change in amyloid. And then there was a dose-related decline at the highest dose. Something like 70–80% of the amyloid was removed from the brain. That's amazing. You just have to say that's amazing. That the drug worked. The drug worked to do what it was intended to do. It removed the amyloid from the brain. It removed the plaques, presumably. The question is, of course, did the people get better? It looks like some of the early data, this is a small trial of 120 people, in 30 in each group roughly. It looks like people stabilized clinically in terms of cognition and function. So it's very tantalizing data, but we've been fooled many times, 100% of the time basically in our field, from going from phase 2 where things look good into phase 3 where drugs failed. Maybe not to have their biology or their chemistry working, but to have a clinical effect. And so now we're going to be doing a trial of thousands of people to see if this really pans out to have a clinical effect, which is the only way that the drug will obviously have a meaningful impact. You heard a little bit also about white matter disease and scans. This is kind of what they look like. And if you talk to a neurologist, and I do this all the time in New York, and I get reports, or from the radiologist, and it's starting to change a little bit now, and they'd say, there are white matter changes in the brain, but they're normal for aging. And that's a failure of translating new research into clinical practice, because we know now that when people have white matter findings like these that are abnormal, that this is a reflective of small vessel disease in the brain, of a vascular contribution to dementia, and probably is related to Alzheimer's disease. And I could tell you about how those models work, but there's little doubt in my mind that Alzheimer's disease affects the deep white matter, and that these white matter changes, if you consider the prevalence of vascular disease in the general population at age 75, it's 80–90%. If you look at, going back, I remember 30 years ago, the pathology, if you look at community-based populations, more than half of people have some sort of vascular pathology in their brain in old age. There were studies coming out of Sweden 20 years ago that showed that the most common cause of dementia in people over 85 was vascular disease and not Alzheimer's disease. So in this sprint towards Alzheimer neurodegeneration and amyloid pathology, which I think is important, we've kind of forgotten that there are blood vessels in the brain, that vascular disease of the brain is very important, and that we can measure it now. We have a biomarker probably, which is not only, which is looking at, as was mentioned earlier, lacunar infarcts, white matter changes that are reflective. And I'll show you in a minute how we are funding a study to see if we can alter these. And the other thing is that through programs like ABLE here in Australia and ADNI in the US and others, we know that the normal brain shrinks about 0.4% a year after about age 50. And the Alzheimer brain will shrink about 4% per year on a volumetric basis. And so we use this marker as a marker of neurodegeneration, the volume changes in the hippocampus or in the ventricles. Here you're seeing what the hippocampus of a healthy brain, and this is, of course, the region where Alzheimer's disease starts and where memory and learning is. And you can see that here's the size of a normal hippocampus, and here's the shrunken hippocampus in Alzheimer's disease. Now, in the past, you really didn't get this information from the radiologist. And if they did, they could only do it on what's called a visual read. But as a result of ABLE and ADNI, the computer software has been developed to analyze this volumetric data and be able to now report it. A company has a program, Cortex, a company called Cortex has a program that's available and approved by the FDA in the US called NeuroQuant. So now when I order an MRI in my practice, I translate the data from ADNI and ABLE, which says that if there's atrophy of the hippocampus, this is a bad sign. It's a sign of neurodegeneration. Now I have two imaging agents that I can use to define a patient with Alzheimer's disease. One is, what's the cause? Do they have vascular dementia? Do they have amyloid in their brain? And number two, is that clinically significant, not just in terms of that very mild memory loss that they have, which might be normal aging, I don't know, but in terms of whether there's neurodegeneration. So the NeuroQuant report that I get out of this, I have patients that come in, they have mild memory impairment. They might have a positive amyloid scan, and then the computer spits out that their hippocampus is less than 1 percentile normal for their age. So they drop off the curve of normal, even including the standard deviation. And that's when I know that those people have something going on, that the brain is shrinking, and that pathology is clinically important, and they have a worse prognosis, that probably in 3 years they're going to be demented. And that helps with care planning, and it's very important. And now we have tangle measures, which I won't go into, but we can actually see the tangles now. And this is going on here in Australia, here in Melbourne, and Victor Billiman and others here are developing these biomarkers. And that's really important because now we can see the dying neurons. We don't just see the extracellular deposits of amyloid. We don't just see the shrinkage of the brain, but we can actually see the dying neurons, which are the tangles that Alzheimer described, and correlate very highly with clinical progression, which means that this could be a surrogate marker in clinical trials. As we treat people, if we can prevent the dying neurons, maybe we'll see a change in the signal on the tau imaging. Now, tau has been a target for a number of years. We've been funding tau targets for years. And I won't go through all of these, but as I mentioned earlier, phosphorylation of tau leads to misfolding, aggregation, toxic oligomers, to breakdown of the synapses and so on. And so there have been several attempts at inhibiting enzymes that are in the phosphorylation pathway. They have low biological validation for being a target in Alzheimer's. This comes from a paper recently published. And all of these drug programs have failed. We have anti-aggregants. We just heard about the failure of tau-Rx, the methylene blue, trying to prevent the anti-aggregants. We've funded microtubule stabilizing agents. Bristol-Myers Squibb had one of these epithiolones in clinical trials. They dropped it. These are very difficult drugs because they come from cancer world. They don't get across the blood-brain barrier, and they're neurotoxic in reasonable doses. And so far they've failed. And now the only thing that's sort of still out there are tau antibodies that are trying to prevent the spread of this toxic tau, sort of along the lines of the way a prion might jump from synapse to synapse and cause toxicity. And some of these are in phase 1 right now. So let's go back to my original funnel. Where are new drugs going to come from? Epigenetics was something I mentioned. It is living on Earth. It is the interface between the environment and our genes. If somebody, such as Audrey Hepburn, suffered severe malnutrition during the war, during the Second World War, and she always claimed that severe malnutrition and depression that she suffered during the war had a lifelong effect on her. It changed her life. Now, you could go to Freud or your clinical psychologist and say, this is psychology. But it's probably not psychology. It's probably epigenetics. It's probably that the machinery, that not a mutation on the DNA, but the machinery that regulates the expression of the DNA was changed. And that's what epigenetics is. It's through oxidation and other translational mechanisms to put molecules on the DNA that change the way the DNA is expressed. Now, Li Wei Sai at MIT showed that epigenetic changes in an animal model actually cause cognitive dysfunction in the neurodegenerating brain. Now, how would this work? Well, here's the DNA wrapped in what's called histones. And when the DNA is wrapped up in the histones, it's not read. It's a read-write phenomenon, and the DNA is not read. But when the histones are acetylated, and that's a small little group that's put on by an enzyme called histone acetyltransferase, the reading frame is opened up, and the genes can be read. Reversely, there are molecules called histone deacetylators that can then wrap up the DNA again. My pointer just went out of battery, but that can rewrap the DNA. So the DNA can be read and written, and not read and written at various times. Now, so with the open reading frame, what's been proposed is that we could develop drugs, and there's about 9 subtypes of HDAC, so again, chemistry is very important here, particularly in terms of side effects. But we could develop drugs that would inhibit the deacetylation and keep the open reading frame. And another group that's put on is a methyl group. It's also put on the histone. And there are other drugs called lysine-specific demethylase inhibitors that could also help to keep this open reading frame and promote synaptic plasticity and prevent the effects of lifelong environment on the brain. And in fact, that's exactly what we're doing. So here's an example of how philanthropy can work. There's a real operating company in Barcelona called Horizon, and they provide services, contract services for drug discovery, and they're a cancer company. And they're trying to develop histone, lysine-specific demethylase inhibitors for the treatment of cancer, because these open reading frames in dividing cells lead to proliferation and tumor formation. So you want to close them down. And what we said to them is we're interested, and they were interested, their chief scientist, Tamara Mace, she was interested in repositioning their lysine-specific demethylase inhibitors for Alzheimer's disease. But their board said, no, you've got to stick to your needing. We have a very expensive program here, and we're not going to do this unless you get external funding and validation from an external organization. So about 4 years ago, we gave Horizon a grant to reposition their cancer program on lysine-specific demethylase inhibitors into Alzheimer's animal models with some new chemistry that they developed to enable these molecules to cross the blood-brain barrier. And I can just tell you that the company just went public in December and is beginning a phase 1 clinical trial, has gone through the IND enabling work, and these lysine-specific demethylase inhibitors that are being developed by this company in Barcelona are now in clinical trials for the treatment of Alzheimer's disease. And here was the announcement in July that they made publicly about ORY 2001 for Alzheimer's disease. Now, there was another company that was actually spun out by Li Wei Tsai out of MIT based on her intellectual property called Rodin Therapeutics, and they were dedicated to developing HDAC2 inhibitors. Again, a difficult chemistry. There are HDAC inhibitors on the market today for the treatment of cancer. So again, repositioning and learning from cancer for the treatment of Alzheimer's disease. This was a preclinical program. We were early investors in the company along with Atlas Ventures, which is a very prominent early stage venture capital group, Johnson & Johnson, and Proteus, which is a contract research organization, to test their HDAC2 inhibitors in an animal model of Alzheimer's disease. And they just announced in January of this year a $500 million deal with Biogen to back their program and bring it into the clinic. We talked a little bit about inflammation. I gave some of the background on systemic inflammation and neuroinflammation. What about translating that biology into new drugs? So what we're doing here is repurposing. There's a lot of drugs on the market for systemic inflammation. The leading drug is Enbrel, or these anti-TNF alpha drugs, okay, that inhibit this cytokine that is a regulator of inflammation. And so Clive Holmes has shown pretty convincingly in phase 2 that people who are treated with this anti-TNF alpha antibody actually have a stabilization of their cognitive function. In fact, he showed in a very interesting study that people that had high TNF alpha decreased in a clinical setting, in a clinical trial, in the same manner that would be looked at as the normal decrease in cognitive function over the course of a year in a placebo group. And the people who didn't have high TNF alpha, who didn't have systemic inflammation, didn't change at all. And he showed that people who were treated with this anti-inflammatory drug actually they had stabilization of their cognitive function. So along with the European Commission, we seeded this trial, and the Alzheimer's Society of the United Kingdom, so partnerships, because nobody, not the Estee Lauder family, nobody can afford these expensive trials, and nobody wants to do this alone. And we want to partner with Yulgilbar across the world, and we're using certolizumab because the original Enbrel antibody wasn't made available by the company, so we're using the second leading drug, and using a biomarker of microglial activation, which is not a great biomarker, and we're interested in developing better neuroinflammatory biomarkers for PET scanning and looking at cognition and PET amyloid scanning to try to decrease. And this has been done before. We've had many anti-inflammatory trials in the past, and they've all failed. But we're not giving up. We've used Motrin and other NSAIDs and so on. Maybe this one will work. But the other thing is that in the brain, there is neuroinflammation. And it's really orchestrated by the microglia, which are specialized cells that are brain-derived. And they have two functions. They have two states that historically have been recognized as M1 and M2. And M1 is the phagocytic state. So these cells are out there trying to find garbage. The other way around. Thank you very much. Glad I saw that. Got the other way around. So M1 is the cytokine production. Thank you. Okay. Stand corrected. So M1 is the state of activation of microglia where they're making a lot of these neurotoxic cytokines, and those are not good for your cells, but they are part of the orchestration of the inflammatory response. And M2 is the state where the cells are trying to be promoted to gobble up this extracellular garbage. And if you look at the plaques and you stain the plaques for microglia, you see all these microglia all around the plaques. In fact, that's what this slide is. You're seeing the glia in the plaques and around the plaques, eating up the plaques. And by the way, I want to mention that a proteomic study of the plaques showed that these extracellular deposits, which Alzheimer called senile plaques, not amyloid plaques, a proteomic study of the plaques showed there are over 450 molecules in the plaques, not just beta amyloid. So they are extracellular deposits of inflammatory debris and cellular debris. And that's not to say that the anti-amyloid strategy won't work, but it's a lot more complicated. So when cells are damaged, they release ATP. And ATP acts as a signal on what are called purinergic receptors on the cell to promote this inflammation. It's a sign of cell damage, and so the microglia come in, and they're activated by the purinergic receptors. And there's different classes of purinergic receptors. Phil Hayden at Tufts spun out a company called GliaCure on the target of P2Y6, which is a purinergic receptor that when it's activated by ATP, right, it promotes phagocytosis, that M1 state. And so we funded him to do a phase 1 clinical trial of an activator of P2Y6 to promote, in that case, microglial activation. And Pablo Peparelli, who was former pharmaceutical company scientist at Pfizer in Italy, he spun out a company called AXAM, which we funded in Milan, to target P2X7, which is a purinergic target that promotes the inflammatory cytokines. And in this case, we want to inhibit the purinergic target for P2X7. And this is a preclinical program. But these are just two examples of novelty in how new drugs are being developed for inflammation. Okay, I have a few more minutes, so I'm going to go through this a little rapidly. We heard a lot about diabetes and comorbidity. About 30% of older people have diabetes. We know from studies that diabetes is a risk factor for Alzheimer's disease and cognitive decline. The brain is highly dependent on glucose for energy. When diabetics drop their glucose below 60, they immediately go unconscious. You give them glucose and they wake up immediately. So mitochondria are very important to neuronal function and survival. The brain is the most metabolically active tissue in the body. 3% of the body weight, 25% of the body's energy utilized in the brain. As I mentioned earlier, John Blass at Cornell has had this theory, a mitochondrial theory of Alzheimer's disease for many years. And in this case, we have been, I won't show you, there are novel targets that we've developed, a company called Metabolic Solutions, which was spun out of Park Davis in Kalamazoo, Michigan, and other companies that we've been supporting to try to address mitochondrial function. But in this case, with Paul Edison at the Imperial College of London, Paul wanted to try a drug called liraglutide, which is a GLP-1 agonist. And in this case, it's repurposing. In other words, it's taking a drug that's already on the market. We know the safety profile. We know the mechanism of action. There's a rationale for using it in Alzheimer's disease. It's been shown in animal models of Alzheimer's disease to reduce beta amyloid. We know it gets in the brain. So all of these factors led us and our reviewers to say, yes, this is worth doing. And we're repurposing liraglutide in a phase 2 trial. How did the foundation do this? We gave him a grant of $750,000, but that's not enough. These trials are often underpowered. What he did was he was able to get Novo Nordisk to donate the drug at a cost of about $1 million, General Electric to donate the beta amyloid scans at another $1 million. The Medical Research Council of the UK came in with a couple $1 million, and the Alzheimer's Society of the UK came in with more money. That was seeded by our $750,000. So now this is a $6 million trial testing liraglutide for Alzheimer's disease. And similarly with Jeff Cummings, a drug called rasagiline, which affects mitochondrial monoamine oxidase B and is probably one of the only drugs ever been shown to alter the course of Parkinson's disease. We're repurposing that at the Cleveland Clinic. There has been a recent failure of a drug, I think from Roche, of a drug called simbragline or something like that, that didn't work. But we're hopeful that rasagiline will help. And I will say one more thing about repurposing. I know that rasagiline is safe. I know it's available on the market. I know that it's used for Parkinson's patients who have neurodegeneration, and I prescribe it. When people come to me and they want hope and they say, doctor, I want you to do something for me, I'm going to take the Aricept, I'm going to take the Namenda, but what else can you give me? And it's a clinical trial of one. It's what we call an N of 1 trial. I can never tell them for sure it works, but I know that I can prescribe this off-label and promote safety, do no harm, and give people a repurposed drug that has a decent rationale for working. I'm going to go through this. I'm almost done. We talked about hypertension and white matter disease. There are angiotensin IV receptors in the hippocampus that are neuroprotective. And some of the most effective antihypertensives out there are angiotensin receptor blockers. And there's good data in animal models, for example, that angiotensin IV blockers can slow down the rate of amyloid deposition. And there's epidemiologic data that people who take these ARBs have a lower risk of Alzheimer's. And so the question from what we call comparative effectiveness is, in people who have hypertension, who want to prevent dementia, and we know that hypertension is a risk factor for dementia. Francois Ferret showed many years ago in the SISTER trial that people who take their calcium channel blocker, nifedipine, had a 50% reduced risk of dementia in 3 years. Dramatic changes, both vascular dementia and Alzheimer's disease. So we're funding 2 clinical trials of angiotensin receptor blockers in people with MCI. One trial of telmisartan is comparing that to an ACE inhibitor in people with hypertension and MCI. And the other trial at Emory University with Dr. Hajar is an angiotensin receptor blocker in people who just have MCI from Alzheimer's disease. And then we're also a part of the NILVAT study, which is testing a calcium channel blocker that's supported by the European Commission. And one of the outcome measures is, can we reduce these white matter findings in the disease? So Sandra Black at the University of Toronto has been a pioneer in the same way that we're using amyloid for anti-amyloid drugs, for drugs where we're trying to look at a biomarker. Does that drug work in small vessel disease from diabetes or from hypertension? What's the biomarker? It's probably these white matter hyperintensities. And so Sandra's developed ways of looking at this on a quantitative computerized basis to give us the sort of biomarker that we need going forward to enhance our clinical trials, to look at white matter disease and the effects of drugs on small vessel. Okay, my last story, I promise, because I know it's 1 o'clock and I stand between you and lunch. Frank, so the most parsimonious, I showed you the funnel, but the most parsimonious way, so regardless of what it is about aging, the most parsimonious way to protect the neurons is to engage the neurons' own neuroprotection pathways that have developed through evolution through receptors like the NGF receptor, where signaling says to the cell, you're too sick, get out of here, go through apoptosis and die. Or, we're not doing so good, but if we signal through P75, which is part of this NGF receptor, we're going to trigger the survival mechanisms of the cell. Now, back 20, 30 years ago, people like Franz Hefty and others tried to use NGF, a big biological molecule, can't be delivered orally, affects the whole receptor, both pathways, no specificity for one pathway or the other, and the drugs failed, caused pain, side effects. What Frank Longo did, he called us up in 2000, and he said, I want to develop a small molecule that mimics the effect of this big neurotrophin and will only hit a specific part of the NGF receptor that triggers the survival pathways that evolution has given us for these neuronal cells. So we paid him money initially to do what's called computational chemistry. We had the receptor, the three-dimensional structure of the receptor up on a computer screen. He bought a computational library, a software library of 2 million compounds. And Steve Massa, his computational chemist, went through a whole year of matching these compounds up with the three-dimensional structure of the receptor, and found 60 of these that fit exactly on the P75 ligand that was the neuroprotective pathway. Now, I'm told that today you can do that same thing in about a week. It took him about a year. He bought the 60 compounds in powder form, and he tested them on cells that were being tested whether the beta amyloid killed the cells, the neurons, and he found 2 of those compounds were actually neuroprotective. So 58 of the compounds, even though they fit in the pocket, they weren't druggable. So these 2 compounds became his leads. We continued to fund him. In 2005, we said to him, Frank, you know, this is coming along. It's getting in towards the commercial space. It's time to start a biotech company because you're not going to be able to run this program on NIH grants. And we need investors. So we were founding investors in this company called Pharmatropics. We continued to fund him to test the drugs in animal models for the IND enabling work to see if it was safe and effective. Just safe and didn't have any toxicology and could meet the regulatory approval of the FDA. The drug was approved by the FDA for human use in 2014. It went through a phase 1 trial, and we've just done a raise of it's about $12 million now to do an early stage phase 2A in Sweden for the first time to see if we can develop these drugs, which are small molecule mimetics of the P75 ligand, to be neuroprotective. Now, there's been a long history of failures in stroke on neuroprotection, but that's a very different model. And so this is an area of science, of drug science, that's been neglected for years because of the failures in stroke. We think that, we're very hopeful, and just to give you the final point of validation, this was on the front page of Time magazine in February, so that's the ultimate scientific validation. I'll skip what we're doing at Rockefeller. We're repurposing an ALS drug that's neuroprotective for the sake of time, Riluzole. So we fund in this valley of death. We're totally focused on drug discovery. I hope I've given you some hope that there are many different ways to develop drugs for Alzheimer's that are parsimonious, that are related to translating the biology of gerontology and aging and living on Earth into new drugs that include anti-amyloid strategies. As you know, we have invested a lot of money. We take a lot of risk and a lot of shots on goal. We hope that they're good shots. We're currently funding 17 clinical trials around the world. We look forward, anybody who's interested in applying to us, just send me an email with your idea and we'll try to help you develop the program and fund it. Of course, we'd have to vet it, so there's no promises. Partnership is the name of how we work. Matching funds. Our founders, our board is totally interested. We partner with drug companies. We partner with other foundations. And I believe that today, from the time when I started in this field in 1980, when people said the only way you could diagnose Alzheimer's disease was at an autopsy, I can diagnose it in my patients in the clinic at the local radiology office. Huge change, and other ways. There are many disease-modifying drugs being tested today. We might actually know by the end of this year if one of them works, the Lilly drug. And we didn't talk at all about prevention, but there's a lot of really interesting information now on prevention, and I'm very excited about the possibility of prevention, not just through drug treatment, but also through lifestyle management. And I'm sorry, I know I went a little long here, and there might not be time for questions, and I stand between you and lunch, but I want to thank you for your attention. Thank you.
Thanks very much, Dr. Fillit. On behalf of the YAF, I'd really like to thank you for taking the time to visit us in Australia and to provide us with your insights and to deliver really what was a sensational and inspirational talk. I think we should take the opportunity to have questions, and perhaps I could ask the first. And it really draws out on a point which is a recurring theme in your talk and which has been drawn out in discussion this morning as well. And perhaps I can put it and couch it in terms of optimism, and that is the segue to success in relation to a partially effective agent. And we're being encouraged to take a reductionist perspective in terms of the A-beta hypothesis. But what you and others have mentioned, I think, is potentially important of comorbidities. And this raises the issue, on the one hand, of phenocopy, on another of multiple pathogenetic mechanisms, on another the issue of redundancy, and another on the issue of compensation. And as a clinical trialist, how do you deal with this in terms of moving forward from a partially effective therapy and the spectre, perhaps, of combinatorial therapies? How does one trial for that? And how does one convince the FDA to deal with that? But there are other examples, for example, in relation to HIV or cancer, where that's the norm. And how do we actually make that transition?
Right. First of all, the regulatory agencies are composed of people whose parents have Alzheimer's disease, or doctors who have possibly even cared for people with Alzheimer's. And what they have said to us is, show us a drug that's effective on a p-value in a classical, clinical trial, and that's reasonably safe, and we'll approve it. And I think, the second question will always be cost and pharmacoeconomics, and let's put that aside for a minute. In terms of, I don't think it's a reductionist approach to say that there's been a lot of focus on beta amyloid because I think the big companies are very risk averse, and it's been the leading target for a long time. And if it does work, it will have a big impact. The comparison, the analogy I like to work is statins. Now, statins are the most successful drug class in history. But if you think about it, what do they actually do? The companies are allowed to say that a statin like Lipitor reduces the risk of heart disease by 30%. But what that actually means from the clinical trial point of view is that in a period of 5 years of a trial, and if you think our trials are difficult, we're running 2,000 or 3,000 people over 18 months. They're running 5,000, 15,000 people over 5 years to get a clinical effect. And the numbers that they come out with are, there are 22 events per 1,000 patients treated over 5 years in the placebo group, and 17 events like heart attacks or strokes in the treatment group. So there's a reduction of 5 events per 1,000 patients treated per 5 years with a statin. And that's, they're allowed to advertise roughly a 30%. But heart disease remains the leading killer in the developed world, despite the tremendous impact of statins. So statins attacking cholesterol have had an incremental effect on heart disease along with lifestyle. But we're still trying to develop new drugs for heart disease along that pathway. So it's not the best analogy. Cholesterol is a very cheap biomarker. It costs $5. The amyloid scans, or the CSFs, are very expensive. But the analogy is there that these diseases of old age are complex. They're caused by multifactorial, and I think all of us, and we just started an initiative on this, we're going to need combination therapy. Most hypertensives today are on 2 or 3 drugs in different pathways. Most diabetics are on 2 or 3 drugs. And most of those drugs are symptomatic, by the way. So we've been discovering drugs for diabetes since the 1920s when insulin was first found by Banting, right? But we can't slow the course of diabetes with any of these drugs, really. It's very difficult. They're not disease-modifying. They're mostly symptomatic. So by trying to analogize, I think that our field will follow the other fields in making incremental, and again, let's take a historical perspective. When I went to medical school and trained in the 70s, I never heard of Alzheimer's disease. So in my professional lifetime, we've literally gone from nothing to where we are today. So there's tremendous hope, and I think that we're going to have incremental, drugs, and the goal is actually quite simple, because unlike other diseases, Alzheimer's is a disease of old age. And so the goal is actually quite simple and I think achievable, and that is that if the average age of onset of Alzheimer's is 75 or 78 or whatever you want in terms of clinical symptoms, and the average time of death for a man is 80, let's say, and a woman maybe 82, by delaying the onset of the disease, the clinical symptoms, by just 5 years, we would reduce the risk of clinical symptoms by 50%. So we'd reduce the number of clinical cases by 50%. So the real question, the realistic question is, would through lifestyle changes and through drugs, which is how we treat heart disease and diabetes, could we delay the onset of clinical symptoms from Alzheimer's disease by 5 years and enable old people like myself to enjoy our grandchildren and die perhaps in the middle of the night from a heart attack? That would be the goal with cognitive function intact. And I think that's an achievable goal.
Thank you. Are there any other burning questions from the audience? Yes.
Yes, our brilliant presentation. What do you say no to?
What do we say no to? We say no to applications that are not druggable. So, one of the first tests is somebody's presenting a molecule that our medicinal chemists say will never be a drug. Drugs that are what's called promiscuous, that have non-specific binding, covalent binding, chemistry. The chemistry is not good. A second thing that we say no to are pathways that we believe are not at all validated. They're too early. And we don't think that those pathways will necessarily help the disease. So we take a priority interest in various pathways. A third thing is that we don't like to fund very early stage work. Most of our programs are medicinal chemistry, proof of concept, proof of mechanism in animals, IND enabling, phase 1, phase 2A. So if somebody comes in with a brand new target and they haven't even developed the assay, the high throughput screening assay, that's too early for us. If they have an assay and we're excited about the target and they want us to pay for a library to screen on the assay, on the target, we might fund that sort of thing. But I think one of the key questions is target validation. And I like to say target validation is a journey. It's not a destination, because you can validate a target in vitro, in vivo, but ultimately, it's the phase 3 that I think validates a target.
One final quick question.
Would you say a little more about what changes in Alzheimer's? Does Alzheimer's occur more commonly in vascular paths, or do you suggest that that's a specific change in the deep mind matter to new Alzheimer's?
It's complicated. If you look at community-based populations, as I mentioned, most community-based populations have vascular comorbidities, diseases that affect the vasculature. I think from a pathological point of view, there is some evidence now that people with Alzheimer's, actually part of the disease might be white matter changes. It might be, we get Alzheimer-described congophilic angiopathy, right? So all of the patients, the vast majority have amyloid, for example. If you consider that a hallmark of Alzheimer's disease, Alzheimer actually described vascular changes as part of the hallmarks of the pathology he saw. There were plaques, there was tangles, there was something he called granulovacuolar degeneration, and congophilic angiopathy. So Alzheimer actually described in the small vessels and in the large vessels of the meninges the deposition of amyloid. So I think it's very possibly an essential part of the disease. but it's also very possibly that these comorbidities contribute to the disease. There was recently an animal model published where they infarcted one capillary in the deep white matter and looked at the columnar segment, what happened to that columnar segment, because the brain is columnar in its organization. And they showed that that entire columnar of neurons through the pyramidal cells in the gray matter was wiped out by that tiny infarction of a capillary in the deep white matter. So the idea was that we think of Alzheimer's as a gray matter disease, neurodegeneration, neuronal driven, and sort of maybe downstream you get the white matter changes. But it could also be the other way around, that ischemia and hypoxia leads neurons to overproduce amyloid or to have a failure of clearance. Ischemia and hypoxia being small vessel disease that leads to amyloid deposition. And I'm reminded of a very classic picture of a capillary, electron micrograph of a capillary in a patient with Alzheimer's disease, which I could show you from about 1976 by a guy named Ishii, where you just see the amyloid pouring out of the perivascular spaces of the capillaries. And we tried to look at whether every plaque actually was associated with a capillary at one point. And it's difficult work. It needs stereology and things like that. But there's the neurovascular unit, right? And so we can't forget that there's over 400 miles of blood vessels in the brain, including these tiny vessels. And then there's the epidemiology, right? So what studies like Abel and ADDF have shown is that people that have white matter disease and Alzheimer's pathology or Alzheimer's signals progress more rapidly. So there's a lot of complex interactions here. But the hopeful message in it is, it says that if you have diabetes or if you have hypertension or if you have high cholesterol, especially in midlife, taking care of that and reducing your vascular risk could also affect the reduction in your Alzheimer's risk.
Thank you. I think we should adjourn for lunch, but ask everyone to be back promptly at 2 o'clock.